Thank you very much, Patrick. And with no further ado, I'll deep into my presentation where I'm complementing and additionally providing information from the big picture in nanomedicine and how we provide solution within the nanomedicine. So I'll be trying to be very practical in what we've been doing. So in chronological time frame, the big picture comes from the 1964 where liposomes were first published. We come here at the far right side. And we were born in 2014, and we've been operational since. And if you were at Scott McNeil's presentation yesterday, they actually started a little bit earlier than us. So they have a lead time which provided a great advantage for the UNCL and the UNCL to actually come together and train and align their activities. So within 2 years of activity, we fastly catch up on the work and the exceptional translation that they did in their framework of the NCI, the National Cancer Institute, as part of the initiative starting in 2004. To quote some of the work that is going to be presented here also from Kathleen Turner, as you can see from 74 to 2015, the number of application of drugs containing nanomaterial has incrementally and exponentially raised quite dramatically with a huge number of application and abbreviating new drug application as reformulation to be increased in 2015 and forward. So we are at the perfect time to create a parallel initiative that can accommodate the European demand of characterization for nanomedicine. This is actually not by chance has been classified and stratified, and liposomes are by far the most frequent, nanocrystal emulsion and nanocomplexes. Cancer is by far the most frequent application, and intravenous injection is by far 63%, by far the greatest. But what happened in Europe? In Europe, we are now seeing something different, and this is a work carried out by one of our colleagues, Robert Gersma at RIVM in the Netherlands, where they actually show the cancer in Europe is by far the largest, with the largest number of products and with the largest number of investigational products. And furthermore, the number of EMA-approved investigational nanomedicine is still standing in the liposome area, in polymer conjugate and in protein nanoparticles. So we are very much aligned between the two worlds, and that's where the US and the UNCL best fit to the demand. As you heard from the opening presentation from Patrick Ussher, you need 2.6 billion euro to develop a nano or a product that goes into the market and a medicinal product. So we are in the space of answer this question, how to support nanomedicine development. And as Patrick has clearly stated, we're here to assist the development and the preparation of dossier that then will go into the application for EMA for new drugs. So what we can offer in practical terms, we provide a preclinical characterization for research and academia, Patrick has clearly stated, but we have in-depth expertise in liposome, in polymeric nanoparticle, in lipid nanoparticle, and in a stream of inorganic particle based on our expertise that we developed through our previous life before UNCL. And through that, we have developed and validated several SOPs and assays which have been aligned with the US counterpart. And from that, we've been achieving our approved stamp from the US to operate in Europe within the same quality and the same characterization profile that is requested and has been developed by the US. So just slightly modified version of the global infrastructure for a global support. This is very much mirrored by the two activity, and I'm going to give you 4 examples of how do we operate and where do we see the value of the UNCL compared to you going out and take your characterization in your own terms, and where we can have a point of strength as a team and as a multi-party activity where we share and we discuss the activity that we carried out within your supported application. The first one is sterility and endotoxicity. We call it the gateway to translation. If this is not met as a requirement, the material will not go into any clinical development. So therefore, with a great and extensive work carried out as a preclinical screening, Munster, as the partner, as indicated by Patrick, assisted by all the other partners, have the duty to screen for endotoxin, but not only to endotoxin, but also for bacterial contamination, which are critically impairing several aspects. It's a parameter for your application. It's by far the largest failure point, as indicated also by Scott McNeil. 30% of their application fail in the endotoxin screening. We require a process development. So if you have found that you have endotoxin, you need to undergo a complete manufacturing revision of your plant. It's the first hurdle for academia. We hate working in sterile condition in academia, but complying with it is one of the must application. And the nanoparticles tend to have also interference at this level. So that's where you learn the first time that maybe your assay is not the right one. And this is where 6 members discussing and developing assay are far more engaging in solving your problem than you going out and reading as much science as you want, because there's actually quite a lot out there that is actually controversy. Furthermore, the next aspect is the physical-chemical characterization, and I say by far the EU and the US have find their point of strength. There is the pillar. It should be actually a joint pillar between the 2 countries in the middle of the Atlantic Ocean, because that's where we bridge the knowledge and we develop assays that are unique, and the communication there is very effective. From that perspective, the first part, the very simple characterization is size distribution, shape, which is basic requirement by imaging analysis, but also surface potential and the coating are critical parameters to be evaluated. And as you know, I borrowed this slide from Fanny Caputo, which is actually in this slide. Life sciences comes in different forms, so therefore we need to accommodate the best delivery of your nanomedicine to be best fit for tackling the medical needs that you are addressing. So let me give you a practical case. We had a sample, which is a liposomal formulation, which had a DLS nominal size of 61 nanometer, and this has been measured under several dilution. But when we did a cryo-TEM, and this is again, thank you to Fanny Caputo, and you can engage with her because she will be at the booth and she will be here in the next 2 days, so Fanny is here. She actually then seen that the particle that look under the 61 nanometer, they weren't actually only particle, and through a further investigation, we actually see that we had a coffee bean-like particle and a spherical particle. So that means there was a bimodal distribution that wasn't detected by DLS. Arguably, we can spend hours discussing about DLS, but this is the standard that also the FDA has introduced into their workflow to get the initial feel and taste of what the particle look like. So having further identification, she can actually distinguish between the 2 size part, and then by asymmetric field flow fractionation, she was also able to actually distinguish between the size range of the spherical-like and the coffee bean-like. And through that was a huge information, because as you can imagine, this is not only surface dependent, there's also volume dependent. That means there could be a different drug loading encapsulating into your liposome. So we move into the chemical composition and drug release, and here I bring you a case of a peptide and a drug present in a formulation. And as indicated by Scott as one of the critical problems, everything works fantastically when you work in deionized water. Your sample is perfect, stable, shelf life, and behave as you're expecting. But the minute you put in a presence of salt, the drug release profile collapse. Or you actually have shift into your structural composition. And this is shown in this simple test that we can actually carry out, where you can actually look at the peptide release in water, in PBS, and culture media at pH 37 degrees. It's pretty outstanding, the difference in the drug release. And when they're combined together, you can actually look at these 2 shifts between the 2 properties and the applicability range in PBS compared to DMEM compared to water. So be careful, and this is one of the critical steps that everybody should try, because if you see precipitation of your particle, then you have to undergo an additional formulation. In vitro cytotoxicity assessment is another requirement in terms of not only ethical approval for the in vivo study, but also to support the evidence that you don't have secondary off-target or secondary target in response from your nanomedicine. And in this particular case, I bring you the example of a cell viability and dose response escalation curve of a inorganic formulation where we could actually see that after 2 runs, we actually have differences in the cell nuclear intensity. And furthermore, on the organelle, we actually see that there is a dose-dependent response. This was matched by the visual imaging that the high-content screening technology is providing us. But I'll leave this one to my colleague, Sven Even, which actually will present far more of what we have developed in this space. In the in vitro, in the immunotoxicity assay, this is quite a broad span of assay that we can actually perform, and this is, again, will actually provide the evidence on how your material will behave when encountering the immune system. And through that, we are evaluating immunosuppression, immunoreaction, and immunocompromising response as part of the complement activation. So in a specific of a liposomal formulation, the formulation was actually developed in a 20 microgram and 230 microgram, and we didn't see any complement activation by running one of the assay. And this was quite strange because we actually have seen that from the physical chemical characterization, there was a quite distinctive difference in the product. So we would have expected something happening. This wasn't confirmed then from the plasma coagulation, which is also being reported in the scheme, where the untreated sample has the same pattern than the treated sample. So quite surprisingly, we see the mismatch between the physical chemical characterization and the immune toxicity response. This brings me to my contribution within the UNCCL with the in vivo in terms of toxicity and pharmacokinetic study, where based on the data acquired and in compliance with the ethical requirement from the European law, we try to design the model for in vitro exploration and to provide you data that will be included into your dossier in small animal models, mainly rodents, but that is not preclusive. We actually mainly focus on maximum tolerated dose and dose escalation, and in some cases into PK, where we can provide you the information that are required in order to identify the safety window where your nanomedicine will be applied. And through that, we will then discuss with the sponsor the aspect of having specific requirements to have GMP quality, have a large volume for sample at high concentration, especially because if you take a dose escalation, you actually will use a huge amount of material because it will be injected in multiple number of animals, as described also by Scott yesterday. So you need grams of material, and not all the academic institutions can actually supply this. Furthermore, this is my question. Are you convinced now that we can provide you some? I see a lot of positive nodding, despite it's not you falling asleep. And I see that the other question is, can you supply us with gram of material? Because if you can do so, well, go onto the website, download the form, and contact Simon, not me. So I hope I deliver the fact that we can tackle the regulatory challenge through the series of brief presentation I've given to you, in terms of providing the big picture of where we're standing and where we can accommodate partially or in full your needs to develop the nanomedicine to go towards the dossier application. Cost is where you get touched particularly, and I was talking to some of you yesterday, where you don't have the cash flow from this perspective, so therefore where we come in free of charge, as Patrick has said, to assist you. And then we actually compile this table for you that will be discussed with EMA, where we actually say, tick the boxes, where you just need to provide this to us. I'd like to thank everybody from the perspective of the joint activity between EU and NCI-NCL. I'll leave to my next colleagues, which Sven Even will provide further insight into the technological aspect, and Simon will slap me in the wrist because I overcommit myself, or I describe, but he will provide you further information on how to apply to the NCL. Thank you very much, and thank you very much for being here at this time of the day.